So welcome everyone, I'm Fabio. So today's webinar, as Andrew said, is about bioinformatics sequence analysis web services. And in this case, I'm particularly describing job dispatcher framework. So just to start the presentation with a quick outline of the webinar. So I'll be describing what job dispatcher is, and then why we should consider programmatic access to the services that we provide at EMBL-EBI. Then I will move to give a quick overview of the RESTful Web Services API. We've got a Swagger user interface page where you can explore the various endpoints of our API. I will then demonstrate the programmatic access to the services and give you an example of a workflow, in this case focusing on common workflow language workflows. And then I will just give you a quick list of examples of EBI resources that use our web services, and leave the presentation with some where to get help and useful links. So you might be familiar with the work that EBI does, and so we provide data resources across different ranges in terms of biology, so from genomes, genes, and genetic variation to the likes of systems biology, molecular structures, chemical biology, and so many more. So today I'm presenting the work that we provide with Job Dispatcher, which is a framework that allows us to provide bioinformatics tools and data to you. And we provide two interfaces to these resources and data, one of which is the web page, the web interface, and the other one is programmatic, which is by the means of clients and HTTP protocols such as REST and SOAP. So as you can see in the left hand side of the page, there's a number of tools there listed, as well as in the middle of the page, there's also data resources. So if you go to this EBI page /services, you can see these listed, and you can search for tools and data resources as well as browse. So not all tools that are provided at the EBI are provided with the Job Dispatcher framework, but a high number of tools that is provided with Job Dispatcher. So Job Dispatcher is a Java application. It's been running for over the last 10 years or so, and it provides basically access to these core bioinformatics tools. These are tools such as Cluster Omega, NCBI Blast, and so many others. And of course, tools like Blast need data to be used. So in this case, we provide also biological databases. So the key feature here is that we provide updated biological databases, and the services, the web services that Job Dispatcher provides allow it to be accessible via programmatic access using the REST and SOAP protocols that I mentioned. And just to give you a quick statistics, as of last year, 2017, we've run more than 140 million jobs with Job Dispatcher. So to give you an overview of the applications that are provided with Job Dispatcher, so you can see here listed, we provide a number of different pairwise and multiple sequence alignment tools such as Cluster Omega, MAFT, Muscle, and tCoffee, for example, as well as sequence similarity search tools such as NCBI Blast, FASTA, and also later added AMR3, HMM scan, and PMR tools. We provide also functional annotation of protein sequences with InterProScan, PFAM scan, and others, as well as other sequence utilities, mostly from the EMBOS package of applications. So this is our tool web page looks like. In this case, for Cluster Omega, you see here at the top of the page a number of links to help and documentation pages, as well as FAQs. You can use those to learn more about the tools and how to use the web services. In the middle of the page here, you can see there's a large text area box where you can paste in your sequences of interest, or alternatively, you could upload a file, for example. I'm not showing in this screenshot, but down in the page, you have a list of options that you can edit if you want to, for example, change the default values for various options and parameters of the tool, and then you submit a job. Once the job is finished, you can see a result page like this, where in this case, we of course see a multiple sequence alignment, which we color in this case, and you can then download the result or link it to use with other tools, or for example, including, for example, third-party tools such as JALView, which is a sequence editor and viewer. So in terms of biological databases, we provide protein sequence databases from UniProt for example, SwissProt, Trimble, UniRef, and UniParc. And we also provide nucleotide sequences from ensemble genomes and DNA, which is the European Nuclear Archive. We provide other sequences from, for example, structures from the Protein Data Bank in Europe and other specialized databases such as Kimball, Intact, and MEREPS. So this is, again, how the web interface looks like for a tool such as NCBI Blast+. In this case, these tools are different from Classal, for example, because here you have a selection of databases that you can pick. And if you expand the sessions that are shown, for example, UniProt clusters, you'd see other databases listed, in this case, UniRef or other databases. Again, you can paste in the sequence of interest in this case, or a sequence identifier, or upload a file and then submit the job. In terms of BLAST, this is how a result page would look like. You'd get a table of sequence hits so for the query sequence that you use, these would be the sequences that BLAST finds to be close to your sequence of interest according to the database that you selected. So now the deal is I would like to try convince you why you should use our web services programmatically. So the web interface is easy to interact with, but of course it doesn't scale. If you want to analyze hundreds or thousands of sequences, going over the web interface is probably not the best you can do. And also another feature that you can have is that you could, for example, install local copies of the software and run tools such as Classal, Omega, and Blast in your own machine, although it would be quite difficult to maintain updated databases, and some of which are pretty large. So this gives you extra flexibility and gives you the potential for you to integrate the web services in your analysis pipelines, as well as resources that you might be developing, for example. So now I'd like to overview the RESTful Web Services API. This is a Swagger interface, user interface page. So there's a drop-down menu there where you click, you can see listed the number of bioinformatics tools that we provide. And for example, in this case, I'm clicking on the Classal Omega menu. So in this case, you see a number of endpoints listed, things like list of available parameters, parameter details, and so on. These are endpoints of the API, and these endpoints are actually shared across different applications. Of course, the output that these endpoints return will differ between the different applications. So just having a quick look over, for example, the first endpoint here listed, list available parameters. So if you run that, you return for Classal Omega a number of parameters that the tool accepts. So, and then in the next endpoint, parameter details for a particular endpoint that you got listed in the previous endpoint, you can know more about it. So in this case, we will get a description of the parameter, as well as default values for the parameter, and so on and so forth. So next, there's a post request endpoint here, /run, where you basically, in this case, you are submitting your query to the server so that the job is run in our servers. So in this case, you are required to pass an email address so that we can contact you in case of, for example, sometime there might be some users trying to kind of overload the servers with a lot of computation. So in this case, we might need to contact you. But generally speaking, there's a relatively there's no many limitations for using the service. There's of course fair usage of the service. So in this case, if you pass a sequence input as well as email, you could then get a job identifier back. And with that job identifier, you then would get to know the status of that particular job. So if you use this endpoint status, you get to know whether the job has been completed or if it's still running, and so on and so forth. The next endpoint here is with job ID as well. You can get to know what the various result types are for a particular job. So in this case, you get to know the various outputs that the tool has generated and that you can then download or use. So now I'd like to, before just going for a quick demonstration of how to use the services programmatically, just to say that we provide web service clients for both REST and SOAP, although REST, Perl, Python, and Java clients are the ones that we are actively maintaining at the moment. And we provide a Docker image that you can basically just use in this case, not needing to kind of install Python, Perl, or Java and the dependencies, so that you can run the clients quite easily in that way. So in any case, the clients, the source code is available in GitHub. You can see the address down there. It's web service clients repository, and of course your contributions are very much welcome if you find any bugs and so on. And of course, you can actually take the source code and modify it for your own use case. So now I'll be doing a bit of a quick demonstration of where to get the clients from and how to run them, and that's the way. So in this case here, I'm showing the web production organization in GitHub, and if you go down to the pinned repositories there, you can see the web service clients repository. And if you go to that repository, there's a lot of information here. There's quite a bunch of descriptions here, how to basically download the clients. So this is a link to the RESTful API. And here you can see Perl, Python, and Java being listed. And it tells you pretty much what you would need to install in terms of dependencies for running Perl, Python, and Java clients. And as I said, we provide also the Docker image that you can use. So in this case, I'll get back to Docker in a few moments. At the moment, I'm going to exemplify how to basically use one of the clients. This one, dbfetch, is a Perl client. I just opened that file and just put it to raw. And now I'll just grab the URL for that particular client. And now I go back to the terminal, and I will just get that client with wget. And I just got the client. I'm going to list, and you can see it there. So if I type perl dbfetch.perl, then I will get a message kind of return from the program, which tells me more about how to run the tool. In this case, dbfetch allows a number of different methods, and I will be using, for example, fetch data to retrieve a database entry. So I can retrieve data for particular, sequences using the sequence identifier. In this case, I'm going to use SP, which is short for Swiss prot, and the name of the protein, and you can see the Swiss prot formatted return. If I type in the format as FASTA, I get the FASTA output. In this case, I'm going to save the output to a file called query.fasta, which I will be using in the next steps to, for example, in this case, using this query sequence, I will be running NCBI blast to get a number of hits from this query against Swiss prot database. So now I'm going to go over to the website again, just to grab the commands to run the Docker commands. So in this case, I'm going to just run the first one, which is docker pull and the name of the image. In this case, running that, I'll get back that I've got the latest version. If there were new changes to the image, I would basically then download the changes. Now, to run NCBI blast client, in this case, I'm going to use the NCBI blast Perl client. If I run just that, as you can see, in this case, I'm using Docker to run the clients. If you downloaded the NCBI blast client, it would be exactly the same output. So these are the number of various parameters that you have, required and optional parameters for the tool, and also a number of general parameters that are shared across all the different clients. So now I'm going to just grab the next command here, just an highlight here to say that we are using the volumes and work directory argument options here for Docker, so that we can basically access the local directory, working directory that we are using at the moment. I'm just copying and paste there from the clipboard my email that is required, and now just copy and paste the rest of the commands. So I will be using type of sequence input to be protein, the database will be Swiss prot, the program itself will be blastp, and the sequence I'll be using the query.fasta file that we just downloaded with dbfetch. I will be setting alignments and scores to be 20 so that I don't get such a huge number of hits from blast, in this case to speed up the process a bit in terms of the next steps in the workflow that I will be showing. And I will be using out format ID. So in this case, I will be only getting one particular result from blast. If I didn't pass out format, I would basically get all the different results that blast returns. So in this case, you can see I got a list of IDs, and I'll just cat the file here. So you can see a bunch of identifiers. These are basically the hits from the blast search, which I can now use to do other things. So what I will be doing now, for example, is using again dbfetch. I'll be getting the sequences in FASTA, and then I will be using that to generate a multiple sequence alignment with class cell omega. So I will be using dbfetch again, now using the file itself. It's ids.txt. I'll be passing here at so that Perl gets the IDs one by one. And if I run that, you can see in FASTA format, I have to say, if you can see that the various sequences were retrieved from with dbfetch. I will just pass those to another file called its.ids.fasta, and it's just doing that. And now just making sure I got the results, I'll just do a head of that file, and as you can see, the results are there. So the sequences in FASTA format are stored in the file. So what I can do now is run class cell omega on this file, which contains these 20 sequences. I'll just copy paste again the Docker command that I use. So in this case, I will be changing BLAST to be a different client. In this case, I will need to use class cell omega client, and I'll be passing help here just to give you an overview of the different parameters. Is similar to what we saw for NCBI BLAST. Of course, in this case, different options because class cell omega uses or has defined different options. So again, passing email, and the sequence that I will be using here is the query its.fasta that I just got from calling dbfetch again. So in this case, I will be using the output format to be class.cell. I could be using FASTA format, for example, as well. And the file that I need from the server, in this case, is just the class.cell alignment. If I didn't pass out format option, I would be getting all the results, and it will be saved as the hits file name as well. So you saw the result there. I will just be adding the file now so that you can see we got a result in class.cell format. This is a multiple sequence alignment that we can use in our analysis. I'll be doing next is to basically generate a phylogenetic tree using simple phylogeny client. This is another tool that we provide. Again, passing email, and the sequence in this case, I can just literally pass the job identifier, so the server job dispatcher knows which output from class.cell to use to generate the tree. I want a tree, that's why I passed the out format tree. And now, as soon as the job is finished, I can show you the tree that we returned. And just cutting that file, you can see this is a Newick formatted dendrogram as one would expect. So as you could see, more or less relatively easily, I ran a number of steps. I could join these steps, for example, in the Unix terminal using pipes or such things, or I could make some custom software to call these tools and to use other tools. But now we can actually use also common workflow language specifications to define workflows, and we are taking advantage of these. So we provide CWL descriptions for the Perl clients, and we provide a sample workflow that you can have a look at. And CWL is a specification for describing these workflows, and it's relatively new, but it's quite powerful. So that's why I would suggest you to have a look on it. So now I'll be showing you how to go to the other repository that we have, where we provide the CWL files, as well as the sample workflow. So if you go to the browser again, so in this case, same GitHub location, in this case, the repository is web service CWL, and we provide here some CWL files for each one of the tools. So these files basically define a command line tool, so in CWL format with the different options and parameters, as well as the outputs and inputs. And I'll just get back to the main page here to show you we've got some description and information how to actually run CWL2, which is the program that runs the CWL files. So now going back to the sample workflow, we provide here in workflows directory some sample workflow, which basically runs the same steps I just shown using the different clients. So the actual workflow file defines a class, which is workflow, and you can see the steps in the workflow are literally NCBI blast step, dbfetch, clustol, and simple phylogeny. Exactly the same steps we just run in the terminal. So now I just moved directories here, and I'll be calling CWL tool with workflows and the sample workflow. In this case, just hitting return will actually run the entire workflow. And as you can see, Docker is being used to run the clients in each one of the steps. And so we are running cluster along step now. And as soon as this one finishes, we should run simple phylogeny to generate the tree. And I think we are there now. So simple phylogeny is just finishing, and soon enough we will get the tree. So all the steps that I ran before are run automatically here using this nice workflow. So I'm just adding the file to see, in fact, we got the Newick formatted tree. So now just moving over with the webinar. So just to quick mention of known resources from the EBI, such as UniProt, Protein Data Bank in Europe, and Ensemble Genomes, which use our web services in their resources. Like they provide blast search engines in their pages that in fact use our web services. Another thing, another resource that uses the web services we provide with Job Dispatcher is into ProScan. So to finish off the webinar, I just like to give you some links here to the tools, to the documentation that we provide, as well as to support. You can also always contact us with your queries. And now some useful links also about the clients and CWL. These are in GitHub, as you can see. And there's the last link here is for the RESTful API. So I would like to finish by acknowledging past and present members in web production that make this work possible. And of course, if you use our services, we would be really thankful if you cite our papers.